Introductory Neuroscience and Neuroinstrumentation, Axonology and Neuronal Biophysics. So here in this session, this is going to be a little complicated, I'm warning you right now, but if you go through it slowly and if you take breaks and try and understand each slide, you'll have a fairly good knowledge of it. So here we are going to consider the properties of axons. So far we have considered action potentials, resting membrane potentials, and how action potentials are formed and how they spread and so on and so forth. But what about the passive properties of the axon? The neuronal biophysics, the electrotonic properties of axon dendrites. Why should we model them? Okay, we model them because using modeling, we can have a pretty good idea and a realistic idea of how it actually works. And to do that, we need to know the different properties which are involved, which cause electrical transmission in axons. Okay. So some background. So as we saw in the earlier lecture on the microscopic anatomy of the central nervous system, neurons have elaborate dendritic trees, like regular trees, arising from their cell bodies and single axons with their own terminal branching patterns. So you have your dendritic trees on top, and then you have your axons going down, transmitting information, and they also have their own trees where they transmit information. There are 5 basic functions. One is the neuron generates intrinsic activity. Second, they receive synaptic inputs from other neurons, axons, cells. So then they integrate all this, they combine the synaptic activity with the intrinsic membrane activity, so it's integrating stuff. Then they encode output patterns in the form of graded action potentials or graded electrotonic potentials, subthreshold potentials. And finally, they distribute the synaptic inputs. Okay, so here you see inputs coming in, and they get integrated in the somatic area. Then you have intrinsic activity of the cell, and then it's encoded, and then you have output in the form of action potentials or graded electrotonic potentials. So a fundamental goal of neuroscience is to develop quantitative descriptions of all these functional operations. 1, we can test experiment-driven hypotheses, and 2, we can do realistic computational models of neurons. When I say realistic, this is compare and contrasted with a very simple form of computational depiction, where you have a neuron which integrates everything coming inside and produces a spike. So these are called spike and integrate neurons. These were earlier ways of modeling neurons, but now we put in details of the dendritic trees, the different conductances, the actual shape of the branches and their diameters, so that computationally it's more requires more resources, but it's more realistic. For example, you have the rat brain on the right, and you have electrodes going into different parts of the brain, and it shows activity, different kinds of activity of neurons or neuronal cells. So here you have regular firing, here you have burst firing, then in the you have intermittent bursts happening in the cerebellum, which we haven't talked about much, and in the midbrain, the thalamus, you have burst modes or fast spikes happening. So there are all these different kinds of neuronal activity which can all be modeled. Excuse me. The first task is to understand how activity spreads. So to do this for a single process such as an axon, it's difficult to begin with. Now when you have branching dendritic trees, it becomes very complicated and challenging. Okay, and then you have interactions between 2 of these trees, it becomes even more complex. And this is one of the main frontiers of neuroscience, how to figure out all the details of what happens in single cells, in cortical columns, and neuronal ensembles. Okay, so here you have 4 possible excitatory inputs which come to a pyramidal cell. Okay, this is a cell in layer 3 of the cortex, and you have cortical afferents, that is inputs coming from other cortical cells. You have spiny stellate layers cells. These are cells in layer 4 which give inputs to layer 3. Then you have other inputs coming from layer 5, and then you have inputs coming from the thalamus. This is a typical sensory pyramidal cell in layer 3. Okay, and the cells can have, as mentioned, can have very different dendritic trees. This is the dendritic tree of a Purkinje cell, which is a pyramidal cell in the cerebellar cortex, and this is in the cerebral cortex, a pyramidal cell. Very, very different, like a banyan tree versus a pine tree. So to model this, we borrowed electrical theory from the engineers, and this is called cable theory and compartment models. So in the 19th century, cables were laid between Europe, England and America to transmit telegraphic signals. Now a cable is basically, it's like a coaxial cable. So you have a central conductor, and then there's insulation, and then it's surrounded by seawater. So the theory for cables can be easily applied to neurons. Very similar maths are involved. And on the right is a famous cable laying ship, the Great Eastern, and that laid the first Atlantic sea cable for telephony between England and America. Okay. Just consider this tree. Okay. So it's mathematically very difficult to apply cable theory to complex branching dendrites. But in 1960s, a great computational neurophysiologist, Wilfred Rall from the United States, he solved this problem by developing computational compartmental models. Okay? And these models have provided a theoretical basis for dendritic function. So combined with mathematical models for generation of synaptic potentials and action potentials, they can provide a complete theoretical description of neuronal activity. And the more data you plug in, the more realistic it is, and faithfully follows what we actually record from the brain. So there are many open source packages which allow even a beginning student like you to explore functional properties and construct realistic neural models. In fact, there's a whole branch of neuroscience, computational neuroscience, where scientists only do this. And for this, you just need a computer and some theoretical background, and you can start doing it at home. Of course, the more you model, the more computation you need. But for starters, a laptop is more than enough to do this. So the program, there are many programs, Genesis, Neuron, etc., which do this. So you just have to learn one properly. So I suggest Neuron. I use Genesis, but I suggest Neuron because there's a lot of documentation available on the internet. And this is Neuron it's from Yale. It's a simulation environment for modeling individual neurons and networks. And it provides tools for building, managing, and using models in a numerically sound and computationally efficient manner. ModelDB is an archive of published models, so you don't have to actually do things from scratch. You can, for example, get a model of a pyramidal cell or a network of neurons from the somatosensory cortex, plug it into your neuron, and then start playing with it, modifying it, as you wish. So you don't have to do it from scratch. Okay. So ModelDB is the database where all the neuron models are there. So Neuron is free; it's an open source program. You have to download it, go through the tutorials, and then use realistic published data from ModelDB and take off. Okay, so now we talk about actually how do you model and what are the assumptions involved. Now, regardless of which modeling software you use, you need to have these facts under your belt. So first point, segments are cylinders. Okay, we treat them as cylinders. So an axon segment is a cylinder. So the electrotonic potential or the local potential, which spreads, that's called an electrotonic potential as opposed to resting membrane or action potential. That electrotonic potential is due to change in the resting membrane potential. Also, it's ohmic, and in the steady state, membrane capacitance is ignored. What we are interested in is the delta or the change in the resting membrane potential. Right? So we have to make some simplifying assumptions. So this local spread of current, also called electrotonic current. So there are two pathways it can take. One is it can go through the axon, and the other is it can go out of the axon via the membrane. So when it goes through the axon, it encounters resistance, and this is internal resistance, or R subscript I. And when it goes through the membrane out, it's membrane resistance, R subscript M. Okay, and here D is the diameter. So more assumptions. Axial current is inversely proportional to the diameter. Okay, so because the resistance is in parallel, sum to decrease the overall resistance, so axial current is inversely proportional to the cross-sectional area. This is straightforward geometry. The other thing is axial resistance, that is the resistance through the longitudinal axis of the axon. It's assumed to be uniform through its process. Hence, the total cross-sectional axial resistance of a segment is represented by a single resistance. So R subscript I is equal to capital Ri, which is a specific internal resistance, divided by the cross-sectional area. So keep it in mind that the units for internal resistance is different from the specific internal resistance. So the units for resistance is in ohms per centimeter of axial length. While Ri is a specific internal resistance, it's ohms centimeter. It's the resistance of a patch. Okay, so A is the cross-sectional area. Okay, so Ri, it's ohms per centimeter, while capital Ri is specific internal resistance, ohms centimeter. Okay. In voltage clamps, the only current is via Rm, so that is going through the membrane. So this permits isolation and analysis of different ionic membrane conductances, as shown in the Hodgkin-Huxley experiments earlier. So membrane current is inversely proportional to the membrane surface area. So for a unit length of cylinder, that's the axonal cylinder, the membrane current I subscript M and the membrane resistance R subscript M are assumed to be uniform over the entire surface. So thus, by the same rule of summing parallel resistances, the membrane current is inversely proportional to the membrane area of the segment, so that a thicker process has lower overall membrane resistance. Thus, R subscript M is equal to capital Rm divided by C, where R subscript M is the membrane resistance of a unit length of cylinder, again ohm centimeter, and Rm is the specific membrane resistance, and C is equal to 2 pi r, okay, the circumference, right? So for a segment, the entire membrane resistance is assumed as concentrated at one point. That is, no axial current flow within a segment, but only between segments. Membrane current passes through the ionic channels in the membrane, as shown earlier, the sodium channels, the potassium channels, the calcium channels, etc. So variations in the channel density and type in different processes and branches can be easily incorporated into the compartment models. So because when we model, we handle segment by segment, so we can change, depending if it's the initial segment where there are more sodium channels, or in the internodal areas with less sodium channels, we can handle all these within our segment models. So the final assumptions are the external medium, the extracellular fluid, if you will, it's assumed to have zero resistance, okay, because of its large volume. And also the resistance of the parts either along a process to the ground or straight to the ground is regarded as negligible, and the potential outside the membrane is assumed to be everywhere equivalent to ground. Okay. We also assume that the ionic concentrations, that the driving force, the EMF across the membrane are constant. So cables. A cable can have different boundary conditions. So it can be infinite boundaries, or it can be a particular length, and it can be closed or open or partial. So for long distance communication, like a cable in the sea, it is considered of infinite length. And these kinds of assumptions carry over to the application of cable theory to long axons. However, if you look at the dendrites, They are very short. So this imposes boundary conditions or the solutions of cable equations which have important effects on electrotonic spread. So suppose it's highly branched, boundary conditions are very difficult to deal with analytically. Like you see a neuron over here, it's very difficult to handle this in a computer. But if you use compartment models, and divide this into, first you use cable models, divide into different cables of different diameter, and each of them is a particular compartment with different resistance and capacitances, then it's tractable, this problem. Okay. So how do you actually do it? How do you construct a compartment model of a passive electrical properties of a nerve cell, either neuron or genesis? So there are discrete steps. The first step is you identify which part of the axon you want to model, which is the segment, what's in there, stuff. Based on this, you abstract this into an electrical equivalent circuit. So you have Cm, which is the membrane capacitance. You have internal resistance Ri, and then you have, of course, the resting membrane potential. Here it's Er, and then you also have Rm. Okay, so we abstract this whole thing and into this, and it actually works pretty well, surprisingly. So suppose you want to do a steady state, spread. So if it's a steady state spread, you can ignore the capacitance and the electromotive force because it's a steady state spread. And if you use a voltage clamp, then even it can be further reduced to only the membrane resistance because everything else is held constant. And instead of Rm, you call it G because call it conductance, which is the inverse of resistance. And finally, the equivalent circuit parameters can be scaled to the size of each segment. One segment may be big, so we can scale that. The next segment may be small, so we can adjust to the size. Okay. So thank you, and in the next session we will get into details of neuronal biophysics.